Welcome to the YouTube live classes again. Now today we will start another new interesting topic of botany, that is molecular biology. Now as you know, the molecular biology syllabus includes the genetic material, the evidences for genetic material, DNA and RNA structure, DNA replication, transcription, translation and gene expression. So, we will cover this whole chapter one after another. So, today we will start the first one, that is DNA as genetic material. So, what we will include here, one is the evidences for genetic material, then we will go to the structure of DNA and RNA, which are mostly considered as the genetic material. So, let us start with, so I am Dr. S.S. SARDAR from RD Women's HSS. So, today we are going to start the DNA as genetic material. So, let us start. So, what are the characteristic feature of genetic material? Now, the genetic materials are like, they must be stable, that means structurally and functionally they must be stable, they must be mutated at a very low rate and they must be capable of expressing themselves. So, these are certain features basing upon which we can prove that which kind of biomolecules are the genetic material. Now, we know that several biomolecules are there like DNA, RNA, protein, carbohydrate, lipid. So, out of this, which one is actually inherited from one generation to another generation, and that compound or that biomolecule in itself is being capable of expressing itself and controlling the activity or the living activity of the whole organism. So, let us have a look again that what are the features of the genetic material. So, one is that it must be stable, it must be capable of being expressed when needed, not that all the time it will be expressed. So, at the time of need it should express itself, then it must be capable of accurate replication, it must be transmitted from parent to progeny without any change, and if any change occurs it must be very slow in nature. So, it must have the scope for slow changes to evolve. So, that means what are the slow changes, and we know that this is the mutation. So, once again it must be any genetic material, it must be stable, it must express itself at the time of need, then it must be capable of accurate replication, and finally it must be transmitted from parent to progeny with slow changes which gives rise or generally in the form of mutation, and the mutation is the basis of evolution. So, the evolutionary process is very slow because of the act of mutation. So, what are the genetic material? Which one is transferred from one generation to another generation, from the parent to the progeny? Now, lot of scientists has actually tried their best to know that which one is the genetic material, whether it is DNA, RNA, protein, carbohydrate or anything else. Okay? So, on that regard, to prove that which one is the genetic material, now many scientists has done many experiments, out of which two experiments are very important, and that proves that the genetic material is nothing else but the DNA. Okay? Now, so DNA is the genetic material, we know, but how we will prove that DNA is the genetic material? So, what are the evidences that proves that DNA is the genetic material and it has all the characteristic feature of being a genetic material? That means it is stable, it is capable of being expressed at the time of need, it is transmitted from generation to generation, and it shows slow changes or mutation, slow mutation. So, the evidences of DNA as genetic material is being proved by two famous experiments. One is Griffith's experiments on transformation, the other one is Hershey's experiment on T2 phase. Okay? So, we will discuss one after another. What is Griffith's experiment? So, we will prove one after another what is Griffith's experiment and what it proves and what is the Hershey and Chase experiment. Okay. So, Griffith's experiment is that in 1928, the British bacteriologist Frederick Griffith conducted a series of experiments using a bacteria known as Streptococcus pneumoniae. So, the bacteria is Streptococcus pneumoniae, and this bacterial experiment was conducted on the model organism, or on which organism? That is mice or mouse. Okay. So, what he was trying? He was not trying to prove that DNA is a genetic material, or he was not trying in that way, but he was trying to create a vaccine against pneumonia using mouse as a model organism. Okay. So, Griffith was not trying to identify the genetic material, but trying to develop a vaccine against pneumonia. Okay. So, what was the experiment? The experiment, or Griffiths conducted the experiment with Streptococcus pneumoniae, which is causing the pneumonia, and he has taken the Streptococcus bacteria or Streptococcus pneumoniae bacteria along with the mouse, and on which he has conducted a series of experiments. Okay. So, what he was trying to do? He was trying to develop a vaccine against pneumonia. So, what is the experiment exactly? Before going to the this diagrammatic picturization, let me tell you that what is the experiment exactly. Now, Griffith in his experiment found that two strains exist. One is a R strain in the Streptococcus pneumoniae, another one is S strain. R strain and S strain. The R strain is, it does not cause virulence, or it is avirulent, whereas the S strain is virulent. What does it mean? Now, this does not cause pneumonia, does not cause the disease, but this cause, or this is the causal reason for causing the disease. So, one is R strain, the other one is S strain. Now, the R strain is avirulent because it is, why it is R strain? It is called as a rough strain. In short, it is a rough strain and it is a smooth strain. The rough strain bacteria does not have a smooth coating or a smooth capsule over there. Okay. So, that is why it appears rough like this. So, the surface is rough somewhat. Okay. And this is the R strain, whereas there is a capsule layer which is present over, there is a capsule layer which is present over the S strain. Okay. So, this is the S strain, and because of the presence of this capsule layer, it appears smooth. Okay. So, because of the presence of capsule layer, it appears smooth, and it does not it does not undergo any sort of enzymatic degradation because of the immunity response of the mouse cells, okay, or inside the mouse body. So, that is why, because of this strong capsule layer, any sort of enzymatic degradation does not affect it, and it is capable of causing pneumonia in the host body. Okay. So, why such? Because there is certain kind of thing which is present inside it, and this is the DNA that we are going to proof. Now what he did, what is the experiment? Now in the first series, now he injected the R strain into the mouse and he found all the mouse in living condition. Okay. In the second series of experiment, he injected S strain into the mouse cell or into the body of the mouse, where he saw that all the mouse who are being injected with S strain, they are dead. Now in the third experiment or in the third series, now he injected the heat killed S strain, okay, into the mouse body, and he saw that all the mouse are alive. Okay. Now here all are alive, this one and this one, and in the fourth one he injected a combination of two. One is the R strain plus the heat killed S strain into the mouse body, and what he found, that most of the mouse, they are found to be dead. So how does it happen? Now as the R strain, it cannot cause virulence, so it cannot cause pneumonia. Now the mouse are all alive. Now in the A strain, as it has the capability of causing virulence and hence pneumonia, so the mouse are dead in nature here, or all the mouse, they are dead because they are being infected by the S strain, and finally what happened? Now finally they are all dead because of the pneumonia disease. Now finally when the heat killed S strain, that means they have raised the temperature to the S strain, or they boiled the S strain colony, and finally they have killed all the S strains by providing heat. So this is called as heat killed S strain. Now when the heat killed S strains are injected into the mouse body, now they were found to be alive because the heat killed S strains does not have the virulence capability, hence it is, they are incapable of causing pneumonia, and finally all the mouse are alive. But in the last experiment, what he did? Now he did that as the R strain and S strain, both of them are not causing any pneumonial symptoms, or they are not being capable of causing pneumonia. So let's inject them together into the mouse body. What will happen? So when he did this, that when he injected both the R strains and the heat killed S strains together, now he surprisingly he found mouse, all the mouse were being injected with this, now they are all dead. Okay, 90% of the cases, now he found the mouse are dead. So why did it happen? So he concluded that there must be, as the S strain causes virulence, so there must be a transforming principle which is hidden inside the heat kill S strain. So that transforming principle get into the R strain and it converts the R strain into the S strain, the living S strain. Okay. Why? Because when he collected the blood sample from the dead mouse, now he found the S strains inside them. So all the dead mouse, they have S strain inside their blood, whereas they would have been, or they have been injected by the R strain and the heat killed S strain. Okay. So that's why they have concluded, or Griffith concluded, that there must be a transforming principle. Transforming principle. He couldn't explain what it was. He just explained that there must be a transforming principle which converts the R strain into a S strain. So, that is why we are getting S strain in the blood of all the dead mice. Okay. So, let us have a look on the diagrammatic presentation. So, when the raw strain non-virulent are injected, now the mouse leaps. Now, when the S strain are injected, now the mouse dies. Now, when the hit kill smooth strain are injected, now the mouse leaps, okay, because they are hit killed. Okay. Now, you see the difference between a smooth strain and a hit killed smooth strain. Now, when the raw strain and hit killed smooth strains are injected together, okay, now he is found the mouse dies, and from the blood sample he got to get, or he got all the mouse who have died, the S strains were present in its blood stream. Okay. So, what he concluded? He concluded that there is some transforming principle which converts the R strain into the S strain, and this transforming principle comes from which one? This comes from hit killed S strain. That means now the DNA which is present, or something which is present here, it comes and it converts this to this. Okay. So, he explained that there must be some transforming principle which was, or which is present inside the hit killed S strain, and this transforming principle from the hit killed S strain comes into the R strain and converts the R strain into a S strain, but he couldn't identify what that transforming principle was. Okay. But later on, with the rediscovery, or later on in 1944, 3 Canadian and American researchers known as Avery, MacLeod and McCarthy, okay, Oswald Avery, Maclean McCarthy and Colin MacLeod set out to identify Griffith's transforming principle, and they found that this is actually the DNA which was the transforming principle. Which was the transforming principle. And how they proved? They proved by, or with the help of some enzymes like DNase, RNase, lipase, protease and what else? Amylase. But they found when the DNase enzyme were or they were tested with the DNase enzyme, now this transforming principle is not being capable of converting the R strain into the S strain. So, they confirmed that this is the DNA molecule which is the transforming principle that converts the R strain to the S strain. Okay. This is one experiment. The second one is the Hershey-Chase experiment. Okay. Now, what it says in their now legendary experiments, Hershey and Chase studied the bacteriophage T2. So, what are bacteriophage? Now, these are viruses which makes the bacteria as their host. So, that attack the bacteria or make them the host. The phages they used were simple particles composed of protein and DNA, with the outer structure made up of protein and the inner core consisting of DNA molecules. Now, Hussey and Chase knew that the phases attached to the surface of the host bacterial cell and injected some substance, either DNA or protein, into the host. These substances gave instruction that caused the host bacterium to start making lots and lots of phases. In other words, it was the phases genetic material. So, let us explain, or let us briefly discuss it. Now, the bacteriophages are the viruses which makes bacteria as their host. Now, they are just nucleoproteins. They are made up of a nucleic acid unit and a protein unit. Now, the bacteriophage, they attach themselves to the outer surface of bacteria, but they inject something into the bacterial cell, either a DNA molecule or the protein molecule. So, out of these two, one is being injected into the bacterial host, and this contains the information or the instruction to produce lots of bacteriophage inside the host cell, and finally it causes the lysis of the host cell and the release of all bacteriophages together in the lytic phase. Okay. So, what did they do? So, as we know, let's have a look on the structure. Now, the bacteriophage, it has a head portion, so they looks like this. Okay. So, they have a head, a tail, and these are the tail fibers. Okay. Now, the head, inside this it contains the DNA strand, and this totally, they all are the protein. This is only the DNA and this is the protein portion. So, togetherly they are a nucleoprotein. So, here you can see. Now, what did they do? Now, for DNA, they labeled the DNA with phosphorus 32 and they labeled the protein with sulfur 35. Why it is so? Because of the presence of phosphorus in DNA molecules only and the sulfur in the protein molecules only. So, they labeled two radioactively labeled atoms like the P32 and S35 into the DNA and protein respectively. Now, what did they do? Now, this P32P labeled DNA and 35S labeled protein, one is labeled with P32, another one is labeled with S35. Okay. So, one is in green color which is being labeled with P32, the other one is red in color or the protein code which is labeled with S35. Now, one batch of phase was labeled with S35, the other one is with P32 and which is incorporated into the DNA, and the S35 is incorporated into the protein code. Now, as we can know that something goes into the host cell, we do not know what goes into the host cell. It might be DNA, it might be protein, we do not know. Now, bacteria were infected with the phages. Well, then after that the culture were blended and centrifuged to separate the phages from the bacteria. Now, what happens? Now, when they are centrifuged, now they found two components, one at the pellet region in one and the other one is supernatant. In the P32 segment inside the bacteria, they found it inside the pellet or in the pellet region, whereas the sulfur 35 they found in the supernatant region. Okay. So, S35 found in the supernatant, that means outside the bacterial cell, because they are not being incorporated into the bacteria, and the P32, they were found in the pellet that is inside the bacteria because it has been incorporated into the bacterial cell. So, radioactivity was measured in the pellet only and in the liquid supernatant for each experiment. Okay. So, radioactivity when it is found inside the pellet in the form of P32 and radioactivity which is found in the supernatant, it is in the form of S35. So, this proves that P32 goes into the host cell whereas S35 it remains outside of the host cell. So, that proves what goes into? It is the DNA strand which goes into the host cell, and this DNA strand is being capable of producing many phases. Okay, so it has been capable of producing many phases after lysis of the cell. So, this proves that DNA goes into the host cell and this is the genetic material which instructs or which has the instruction to produce many phase molecules because of by utilizing the synthetic machinery of the host cell, and it is only the DNA molecule which has the instruction to produce the whole bacteriophage comprising of both the DNA molecule and the protein coat. Okay. So, this again proves that DNA is the genetic material. Once again, the evidences for DNA or of DNA as a genetic material was being confirmed by two famous experiments. One experiment is on Griffith's experiment on transformation, but he found that there is some transforming principle which converts the heat killed or which converts the R strain into a S strain by the help of the heat killed S strain. And it was confirmed later on. What is that transforming principle? It was confirmed later on by Avery, MacLeod and McCarthy in 1944. And then the Hershey-Chase experiment, it also proves, Hershey-Chase experiment on bacteriophage, it also proves that DNA is the genetic material. Nowadays, we cannot say that DNA is the genetic material universally because there is presence of certain RNA viruses which has RNA as their genetic material, but what they do by the help of reverse transcriptase enzyme, the RNA is being converted to a complementary DNA molecule which again takes over the synthetic machinery of the host cell, creating a lots of this viral cells or the viral coats or the viruses, the RNA viruses. However, most of the organisms or all the organisms except few viruses or except some viruses, now DNA is the genetic material. Okay? It has been proved by Griffith and Hershey-Chase. So, this is all about the DNA as the genetic material, but additional research by many scientists including Chargaff, Watson, Crick and Franklin, it led to the discovery of DNA structure and clarifying how DNA can encode large amounts of information, and this forms the basis of molecular biology, because all sort of information that is encoded within DNA, it is being expressed into the form of mRNA by the process of transcription, and the mRNA is converted into a molecule of protein by the process of translation. Okay. So, this forms the basis that whatever is being expressed, it is from the DNA molecule and finally it is being converted to a protein molecule. So, before going to that, we should know what is the structure of DNA molecule. Now, what is DNA? DNA is made up of, or it is made up of molecules known as nucleotide, or in short I can say that DNA is a polymer of, this is a polymer of what? Nucleotides. So, DNA is a polymer of nucleotide. Okay. Now, what is a polymer? Now, as you know, a very famous example is polythene. Now, the polythene bags in which we are carrying many different goods or something, the vegetables and all we are carrying into our home. Nowadays the polythene is banned, or it is not being it is being advised not to use the polythene in a much larger amount. However, the polythene is a polymer. Now it is a polymer of what? This is a polymer of ethylene molecule. So many ethylene molecules joined together forms the polyethylene, or in short this is known as polythene. Similarly, DNA is a molecule which is being formed by joining of many molecules of nucleotides. So in short we can say that DNA, which is otherwise known as a nucleic acid, DNA is a nucleic acid, whether DNA or RNA, they are called as nucleic acid. Why? Because they are found inside the nucleus, and at pH 7 they act as an acid. That means at pH 7 they furnishes H plus ion into the solution. So that is why they are called as, or they shows acidic properties, and as they are found inside the nucleus, or they were discovered inside the nucleus, they are called as nucleic acid, both DNA and RNA. So the DNA or RNA, whatever it may be, now the nucleic acid is a polymer of nucleotides. So we have to find that what a nucleotide, or how a nucleotide is made up of, or what comprises a nucleotide, and what are the different components of a nucleotide. So let me tell you that this is simply just like a string comprising of many beads, and each such bead, okay, so each such bead are the nucleotides. Now there are two polar ends, one is a 5 prime polar end and the other one is a 3 prime polar end, and these are all nucleotides. Okay, two consecutive nucleotides, either this one, this one, this one, like this. Now two consecutive nucleotides are joined together by a bond known as phosphodiester bond. Okay, so you take any nucleic acid, whether it is DNA or RNA. Now both of them has a 5 prime polar end and a 3 prime polar end. Now from the 5 prime end, many nucleotides are joined together, all such bits, let us consider that all such bits are nucleotides, they are joined together by the help of a bond between them which is known as phosphodiester bond. So this is the basic structure of any nucleic acid, whether it is DNA or RNA. Now the DNA molecules has 3 components, or in short we can say a single nucleotide, a single nucleotide, a single nucleotide, that means a single one like this, okay, a single nucleotide comprising of 3 components. So one is a nitrogenous base, the other one is a pentose sugar, and the third one is a phosphate group or phosphoric acid. So what are the 3 components of a nucleotide? One is a nitrogenous base, pentose sugar or carbon sugar group or pentose sugar, and third one is phosphoric acid. Okay. So let's have a look on the nitrogenous base part, the pentose sugar part and the phosphoric acid. So structurally the nitrogenous base are of 2 types. One is a purine base, the other one is a pyrimidine base. Now the purines are double ring structure. They are double ring structure, whereas the pyrimidines are single ring structure. Okay, they comprises of a single ring, they comprises of a double ring, and they are being numbered as this. Now they are cyclic in nature and being composed of carbon nitrogen in an alternative form. Okay, so the purine and the pyrimidine. Now what are the purine nitrogenous base? Adenine and guanine. These two are the, in short they are called as A and G. These are the purine bases, and the single ring are, or the pyrimidines are thiamine, short T, and uracil, U, and cytosine, C. So adenine and guanines are the purines, thymine and cytosine are the pyrimidines which are found in DNA. So the thiamine is present in DNA nucleotides, whereas the uracil is found in RNA nucleotides as a nitrogenous base. Okay, so the nitrogenous base thiamine is present in DNA and the nitrogenous base uracil is present in RNA. This is the only difference. Whereas both the RNA and DNA molecules or the nucleotides, they comprise of the rest three like cytosine, adenine and guanine. Okay. So, let us have the look on their structure. Now, this is the structure of the nitrogenous base guanine. Okay. This is called as, or this is the structure of adenine. Okay. The adenine are nomenclatured as 6-amino purine. Okay, they are nomenclatured as 6-amino purine. That means an amino group is present. You can see a 6 group is, an amino group is present at this end in case of adenine molecule. Okay, so you can see it here, and in guanine the amino group is at second position and the carbonyl group or a carboxyl group is present at the 6th carbon. Okay. So, that is why it is called as 2-amino 6-oxopurine. Okay. 2-amino 6-oxopurine. Now, this is the structure of cytosine, this is the structure of uracil and thymine. Well, thymine is 2,4-dioxo 5-methyl pyrimidine. These are all purine, these are all pyrimidine, and uracil is only 2,4-dioxopyrimidine, and this is 2-oxo 4-amino pyrimidine. So, these are the structure of this 5 nitrogenous base: adenine, guanine, cytosine, uracil and thymine. Now, how do they base pair? Now, they form, generally the adenine and thiamine, they form 2 hydrogen Now, the adenine and thiamine, they form 2 hydrogen bonds between them, whereas the cytosine and guanine, they form 3 hydrogen bonds between them. Okay, and this is the basis how they form. These are the hydrogen bonds, and that is formed between the thiamine and the adenine, whereas these are the 3 hydrogen bonds which are being formed between cytosine and guanine. So, here 2 hydrogen bonds are formed, here 3 hydrogen bonds are formed. Now, what is the basis of the formation of hydrogen bond? Hydrogen bonds are weak bonds and it is formed between 2 atoms which has a greater electronegativity difference. Okay. So, it is generally formed between atoms like N and H and O and H. Okay. So, you can see here N and O, here it is being formed and here it is being formed between N and N. So, the hydrogen bonding is formed, as it is a weak bonding, it is formed 2 hydrogen bonds between T and A and 3 hydrogen bonds between G and C. So, next component is the pentose sugar. Now, pentose sugar is of 2 types. One is a ribose sugar, another one is a deoxyribose sugar. Now, pentose sugar means, now there are 5 carbon molecules or 5 carbon, this is a 5 carbon skeleton, this is in the form of a ring, okay, and the 5 carbons are present at 4 different corners of the pentose ring, whereas the last carbon is present outside the ring. Okay. So, in case of ribose, now you can see the difference. In case of ribose, a hydroxylic group is present at the second prime carbon of the pentose sugar. In case of deoxyribose, this hydroxylic group is absent. Okay. So, instead of the hydroxylic group, there is a H atom here at the second prime carbon. So, what it lacks? It lacks an oxygen atom. So, a single oxygen atom is being not present here. So, that is why it is called as deoxyribose. That means it is less of one oxygen as compared to the ribose, as compared to the ribose sugar. Okay. So, next is a phosphoric acid or phosphate group. Now, as you know the structure of a phosphoric acid. So, this is the structure. Okay. Now, now we know that a nucleotide comprised of three components. One is a nitrogenous base, one is pentose sugar, the other one is phosphoric acid. But these three combine in a unique way to form the nucleotide. So, first of all, a nitrogenous base, it combines with a pentose sugar, okay, to form a nucleoside. Now, the nucleoside combines with the phosphoric acid to form the nucleotide. So, let us have a look on the diagram. Now this is the nitrogenous base. It combines, when it combines with the pentose sugar. Now, this forms the nucleoside. Okay. Now, this is the the nucleoside, here this is the nucleoside, when it combines with the phosphate group. Now, this becomes the nucleotide. Okay. Now, this is the structure of a nucleotide. Now, this is adenine nitrogenous base. Now, that forms a glycosidic bond with the pentose sugar or the deoxyribose sugar. Here, this is the nitrogenous base component, that is in the light yellow. In the sky blue component, this is the deoxyribose sugar, or any sugar we can take, either deoxyribose or ribose sugar. Now, this two forms what? The nucleoside. And the nucleoside, when it combines with the phosphate group, now this becomes a nucleotide. Okay. So, this is the complete unit of a nucleotide, and this is being what? This is being called as adenosine. This nucleotide is called as adenosine monophosphate. So, the nucleotides are read as like this. So, they are always what? Nucleoside monophosphate. In case of adenine base, it is adenosine monophosphate. Now, let us suppose here there would be a G. Now, when it is a guanine, now this is called as guanosine monophosphate. Now, when there is a T base here, now this is called as thymidine monophosphate. Now, these are for ribose sugar. Now, for deoxyribose sugar, this is something like deoxyribose adenosine monophosphate and guanosine monophosphate, like that. Okay. Now, when there is a C, it is called as cytidine monophosphate. If it is a U, it is called as uridine monophosphate. Okay. So, the nucleotides are named as they are nucleoside monophosphate or diphosphate or triphosphate. For example, ATP molecule, this is a nucleotide. What is the full form of ATP? This is adenosine triphosphate. So, adenosine triphosphate is a nucleotide because adenosine is the nucleoside which is combined with the three phosphate groups forming a nucleotide. Okay. Now, this is the complete structure of adenosine monophosphate nucleotide. Now, here when adenine nitrogenous base combines with the pentose sugar, this becomes adenosine nucleoside. Now, when it combines with a phosphate group, it becomes adenosine monophosphate or a nucleotide. Now, let us suppose here if I will go on adding, okay, if we will go on adding another phosphate, now this becomes what? Adenosine diphosphate. Now, if it is being combined with another phosphate group, okay, now this becomes adenosine triphosphate like this, but they are all nucleotide. Now, we have come up to a nucleotide group. Now, from a nucleotide, now how a dinucleotide will be formed? Now, a nucleotide combines with another nucleotide, now it forms a dinucleotide like this. Now, how it forms? Now, a dinucleotide is formed between the hydroxylic group to the phosphoric acid. Okay. Now, in this way a dinucleotide is formed, and here it always maintains the polarity from 5 prime end to 3 prime end. Now, from a dinucleotide, that means when two nucleotides join together, it forms a dinucleotide. Now, with this dinucleotide, if another nucleotide is joined together, it forms a trinucleotide. Next, a trinucleotide, when it is joined with another nucleotide, it forms a tetranucleotide. So, likewise, when n numbers of nucleotides are joined together, now they forms a polynucleotide. So, this is the structure of a polynucleotide, and how they are being formed. Now, you see, now there is always a phosphodiester bond is formed between the 3 prime hydroxylic group to the 5 prime phosphate group. Okay. So, always you see, this is the phosphodiester bond which is formed between the 3 prime hydroxylic group to 5 prime phosphate group of the next nucleotide. So, this is the previous nucleotide and this is the next nucleotide. Okay. So, always there is maintained a 5 prime end towards one terminus and a 3 prime end towards the last terminus or towards another terminus. Okay. So, this is the structure of a polynucleotide, but in case of DNA, now there are not a single strand of polynucleotide, rather 2 strands of polynucleotides are there. So, this is the structure where you can see that these are the sugars, these are the nitrogenous base and these are the phosphate groups which forms the phosphodiester bond. Now, this is a single polynucleotide, okay, and this is another polynucleotide. Now, in DNA molecule, two polynucleotides are present which forms complementary base pairing between A and T and C and G, or T and A or G and C. Okay, whatever may be the nitrogenous base sequence, accordingly another complementary polynucleotide is created and they form a complementary base pairing with each other. So, one strand runs from 5 prime to 3 prime in this direction, whereas the other strand runs in the opposite direction from 5 prime to 3 prime. So, that is why the DNA molecules are called as anti-parallel in nature. Anti-parallel DNA strands, or the 2 strands of DNA molecules, they are anti-parallel to each other, okay, but both of the strands are polynucleotides. Now, next, if we will consider the structure of RNA being a nucleic acid, being a polymer of nucleotides, okay, this polymer comprises of a single strand, not 2 strands. So, you take any of the RNA like mRNA, tRNA or rRNA. Now, all the RNAs, they are always single strand in nature, okay, they are all single stranded. Now, regarding the physical structure of DNA, now we have come across the chemical structure. Now, these are the nitrogenous base, the pentose sugar and this the pentose sugar and the phosphoric acid. They all are the chemical components of the DNA. First of all, they form nucleoside, then a nucleotide, then many nucleotides join together to form polynucleotide, and these polynucleotides are the chemical structure of DNA. But apart from the chemical structure, now it has a physical structure. Now what is that physical structure? The physical structure of DNA is what? This is the helical structure of DNA, which was being discovered by Watson and Crick. Okay? So this is the physical structure of DNA, which is helical in nature. Now if we will put on a structure of this, now mostly Watson and Crick, they discovered the bDNA form of the DNA. Now what it says, now this DNA is double helical, or this is double helical in nature. Now the diameter is 20 angstrom, okay, and a single 360-degree turn was taken, or one complete turn was taken in 34 angstrom units. Okay. So that means when it takes a complete 360-degree revolution or complete 360-degree rotation, now it covers a distance of 34 angstrom. Okay. And there are some minor groups and major groups present in this helix, and the backbone is sugar phosphate molecule or sugar phosphate backbone. The backbone is being formed by the sugar phosphate, and the nitrogenous base, they are projected into the helix, and when the nitrogenous bases come closer towards each other, that means when A comes close to the T, now it forms 2 hydrogen bond. Now when a C comes closer to the G, now it forms 3 hydrogen bonds. So likewise, the nitrogenous bases, they form complementary base pairing. Okay, and the distance between two consecutive nitrogenous bases, okay, between two consecutive nitrogenous bases are 3.4 angstrom. So it confirms that the 34 angstrom unit, it comprises of 10.5 base pairs, okay, or this distance cover 10.5 base pairs of the DNA molecule. So this is the structure, physical structure of a DNA, and particularly this structure is for the B form of DNA that was discovered by Watson and Crick. Besides this B form of DNA, we have 2 other forms. Now the different forms of DNA, so the major one is the bDNA. Apart from that, we have aDNA and jDNA, and they differs by their characteristic feature or the structural parameters. Now for example, the aDNA is a right-handed helix, it is a right-handed helix, whereas bDNA also right-handed, whereas jDNA is left-handed. jDNA is left-handed. So these are the comparative form of all the 3 DNAs, aDNA, bDNA and zDNA. Now out of these 3, zDNA is left-handed. Now the residue per helical turn is 11 in case of aDNA, 10.5 in case of bDNA and 12 in case of zDNA. Now the diameter of the helix in case of aDNA is 23 angstrom, in case of bDNA is 20 angstrom, and in case of jDNA it is 18 angstrom. So the pitch length of the helix is 2.82 for aDNA, 3.4 angstrom for bDNA and 44.4 angstrom for aDNA. Okay. Now these are the different features of the 3 DNA, A, B and Z DNA. So this is the structural difference. Now this is the bDNA, which is the major form of DNA, whereas aDNA is somewhat different and jDNA is a left helical structure. So this is all about the DNA as genetic material and the structure of DNA. Okay. So in the next class we will go for the DNA replication part, but before that we will also have a look on the structure of RNA too. So have a good day.